Hello, welcome once again to our channel Bioholic with your Friday educator Olivia. So today is the third part of the locomotion and movement. And here I will tell you about the mechanism of muscle contraction. This is the most important part of the chapter Locomotion and Movement. So watch this video attentively till the end. Not wasting more time, let's start today's class. In the last class, I told you about the contractile proteins, actin and myosin. Each actin filament is made up of two F or filamentous actins helically wound to each other, and each F actin is a polymer of monomeric G or globular actins. Two filaments of another protein, tropomyosin, also run close to the F actins throughout its length. Troponin, which is another complex protein, is distributed at regular intervals on the tropomyosin. In the resting state, a subunit of troponin masks the active binding sites of myosin on the actin filaments. Here you can see the tropomyosin, troponin and G-actin molecule present in the thin filaments. Okay? On the other hand, each myosin filament is also a polymerized protein. This is also called as thick filament. Many monomeric proteins called meromyosins constitute one thick filament, and each meromyosin has two important parts, a globular head with a short arm and a tail, the former being called the heavy meromyosin or HMM, and the latter, that is the tail, is the light meromyosin or LMM. The HMM component, that is the head and short arm, projects outwards at regular distance and angle from each other from the surface of a polymerized myosin filament and is known as cross arm. The globular head is an active ATPase enzyme and has binding sites for ATP and active sites for actin. So this is the actin binding sites and this is the ATP binding sites that is present in the head of the myosin. And here you can see the arrangements of the actin-myosin complex. Thus, the myosin head gets attached to the actin filament. Okay? Now, the mechanism of muscle contraction. The process of muscle contraction occurs over a number of key steps, including number 1, depolarization and calcium ion release, then the actin-myosin cross-bridge formation, then the sliding mechanism of actin and myosin filaments, and lastly the sarcomere shortening. So let's look into the first step, that is the depolarization and calcium ion release. An action potential from a motor neuron triggers the release of acetylcholine into the motor end plate. What is motor end plate? The junction between a motor neuron and the sarcolemma of the muscle fiber. So due to the depolarization, the sarcoplasmic reticulum releases the stores of calcium ions, and the calcium ions play a pivotal role in initiating muscle contraction. Now the step 2, actin-myosin cross-bridge formation. On actin, the binding sites of the myosin heads are covered by a blocking complex, that is by the troponin and tropomyosin. But calcium ions, after get released, they bind to troponin and reconfigure the complex, exposing the binding sites for the myosin heads. And the myosin heads then form a cross bridge with the actin filaments. Next step is the sliding mechanism of actin and myosin. Here, ATP binds to the myosin head, breaking the cross bridge between actin and myosin. Then ATP hydrolysis causes the myosin heads to change position and swivel, moving them towards the next actin binding site. The myosin heads bind to the new actin sites and return to their original conformation. This reorientation drags the actin along the myosin in a sliding mechanism. That's why this is known as sliding mechanism of actin and myosin. The myosin heads then move the actin filaments in a similar fashion to the way in which an oar propels a boat. And lastly, the step 4, sarcomere shortening. The repeated reorientation of the myosin heads drag the actin filaments along the length of the myosin. As actin filaments are anchored to Z lines, the dragging of actin pulls the Z lines close together, shortening the sarcomere. And as the individual sarcomeres become shorter in length, the muscle fibers as a whole contracts finally. To make you understand better, here I have provided the steps in video format. So in the step 1, you can see calcium ions comes, then their conformational change occurs that exposes the binding sites and myosin heads attach to the binding sites. And in the next step, the myosin heads bind to the ATP and then ATP hydrolysis causes the myosin heads to change its position, and then it goes back to its normal stretch and move forwards and binds to the next active binding sites. Now, thus, the actins move forwards towards the Z line. Again, ATP binds to it, and ATP hydrolysis causes detachment of the cross bridge, and the myosin heads gets reactivated. This process occurs again and again. Here I have provided the steps of cross bridge cycle in a chart form. In the first step, the active site on actin is exposed as calcium binds troponin. Then the myosin heads form a cross bridge with actin. Then during the power stroke, the myosin head bends and ADP and phosphate are released, and a new molecule of ATP attaches to the myosin head, causing the cross bridge to detach. And lastly, the ATP hydrolyzes to ADP and phosphate, which returns the myosin to the cocked position or to its normal position. The myosin head thus moves forward the M line, pulling the actin along with it. As the actin is pulled, the filaments move approximately 10 nanometer towards the M line, and this movement is called as the power stroke. Okay? And the process continues till the calcium ions are pumped back to the sarcoplasmic cistern, resulting in the masking of actin filaments. This causes the return of Z lines back to their original position, that is, the relaxation after contraction. Okay? Here once again you can see the sliding mechanism, and you can see how the sarcomere gets shortened. So the mechanism of muscle contraction involves the following steps: initiation of muscle contraction through the CNS and motor nerve that reaches in the neuromuscular junction, and then acetylcholine is released. That's why there is a potential development. Nextly, calcium release in sarcoplasm that triggers the head of myosin active open location of actin bridge formation between them, and then attached actin fibers pulled towards the middle part of the A band. then pulled inner side Z line, which is also attached within the actin so, and the sarcomere becomes shortened. Muscle contraction by muscle release the ADP and PI, and then muscle relaxation occur. And you should remember one important thing, that is only in absence of ATP cross bridge formation takes place, and the cross bridge breaks in presence of ATP. So that's all about the mechanism of muscle contraction. If you have any queries, you can ask me in the comment section, or you can also directly mail to us. I will definitely try to clear all your doubts. And the self-assessment test link is provided in the description box. Go and check that. Thanks for watching. See you in the next class, where I will describe about the skeletal systems of human being. So see you soon. Till then, stay tuned with Bioholic.